I will be focusing on a research paper that discusses transcription factor binding kinetics in relation to nucleosome presence. This paper investigated that transcription factor suppression and dissociation is an effect of nucleosomes. This research discovered that nucleosomes influence transcription factors, more specifically LEX-A. Previous studies investigated the same transcription factor and found that LEX-A bound to its target sequence within a partially unwrapped nucleosome, even though the concentration to bind 50% of the nucleosome was 100-fold higher relative to its binding to duplex DNA. They also state that many factors influence LEX-A occupancy. The hypothesis in the study states that the nucleosome suppresses transcription factor binding, but it also influences transcription factor dissociation. These transcription factor binding and dissociation dynamics were quantified using SMTIRF microscopy. By using protein-induced fluorescence enhancement, the binding of two transcription factors to their target sites were detected, LEX-A and GAL4. This helped determine that nucleosomes not only suppress transcription factor binding, but also enhance the rate of dissociation.
As shown in figure 1A, how transcription factors work is that they target specific genes by binding particular DNA sequences with high affinity. And this can be measured by calculating the dissociation constant, or KD, as shown in 1B and 1C. This dissociation constant can be calculated with either single molecule or ensembles of molecules together. In figure 1D, the detection of the binding of LEX-A and GAL4 was performed with protein-induced fluorescence enhancement, or PIF, which detects protein binding by attaching a fluorophore, such as CY3, adjacent to the transcription factor target sequence for single molecule experiments. Experiments were also performed, as shown in figure 1D and figure 1E, to quantify the ensemble binding and dissociations of LEX-A and GAL4.
Shown in this figure here, this is the investigation of the LEX-A binding and dissociation. This was done in this previously and shown that LEX-A had movement of transcription factors based on the nucleosomes, and they just recreated it in the study just so that they could use it as a basis of comparison for GAL4.
So this is figure 5 of this paper. And as Riley just said, the authors of this paper want to see how other transcription factors had affected nucleosome dynamics, and therefore they tested GAL4, which is just another transcription factor that binds the nucleosome. So as seen on the figure on the left, as GAL4 concentration increases in this in descending order, so GAL4 at 0 picomolar had high FRET efficiency, and as the GAL4 concentration increased, FRET efficiency decreased, which is mirroring the LEX-A FRET changes. All right, and then in the next graph, we have the relative change in FRET, and this is also going to be the difference between an ensemble and single molecule studies, and we can see that as the GAL4 concentration increases, the relative change in FRET decreases.
Figure 6, so we know that nucleosomes have some effect on transcription factor binding and dynamics. Here they propose a model for competitive binding sites between nucleosome wrapping and partially bound transcription factors binding to the DNA site. So on top right here, they show that transcription factors could partially dissociate. This releases the DNA target site and either rapidly binds again or is fought out by the nucleosome rewrapping. On the right is the model. You see the transcription factor successfully binding, while on the left you see the competitive binding of the nucleosome rewrapping and not allowing access to the DNA site anymore. If the transcription factor can no longer fully bind to the DNA site once again after partially dissociating, is released completely and the nucleosome rewraps completely. They're able to find that the rate of transcription factor binding to a target site within the entry-exit region of the nucleosome is reduced by over 2 times compared to the duplex DNA, while the rate of dissociation is increased by 3 times. This study proves that just not LEX-A is affected by the nucleosome dynamics, but other transcription factors as well. Further investigations to be into which other transcription factors are affected in what manner, and additionally, what other types of DNA binding complexes have similar dynamics to the transcription factor studies shown here. If anyone has any questions, please let us know. And here's the paper that we worked on. Thank you.